Okay, everyone, we're going to get started for today. Thank you for coming. Thanks for joining us if you're on WebEx. Welcome to the Conservation Lecture Series. We have a website now that you can visit and see what lectures we've got coming up. You can also see videos of past lectures. And if you have any questions about the lecture series or about the website, you can contact me. Just to let you know what we've got coming up, we have one lecture per month scheduled through October. So we're going to be having white abalone in July, rearing salmon in the Yolo bypass in August, California red-legged frog in September, and Townsend's bigger bat in October. And today, we're very lucky to have a lecture on Amargosa Vole, and we're going to turn it over to Dr. Deanna Clifford, who's going to give you the introduction for our speakers today.
Good afternoon, and thanks for coming, both people that are in the audience and our virtual guests online. It's really my pleasure to introduce two amazing ecologists who I've had the pleasure of working with. The first who will be speaking today is Dr. Janet Foley. She's a disease ecologist, a veterinarian, and a small mammal specialist, and a professor at the UC Davis School of Veterinary Medicine. She earned her Doctor of Veterinary Medicine degree at UC Davis in 1993, her PhD at Davis also in 1996. Her research focuses on the ecology of disease in complex communities, particularly vector-borne diseases and vector-borne transmitted pathogens, and the impact of disease on small mammal populations. And so Dr. Foley is going to lead the first part of the lecture, and then it will roll smoothly into Dr. Rob Klinger's part of the lecture. And the work is really complementary, and so the two of them have really been working together to give you today what I feel is a really thorough picture of the current state of the Amargosa vole. Dr. Klinger is an ecologist and also a UC Davis graduate with the USGS. He has special interest in community and population ecology and dynamics in plant-animal interactions. He's worked on multiple projects with small mammals, including a large project in the alpine zone of the Sierra Nevada. Also has some collaborative department there, looking at climate, fire regimes, vegetation dynamics, and invasive and non-native plants. So he has a lot of experience in the Mojave ecoregion, and these were the two people we needed to bring in at the time when we were having challenges with the Amargosa vole, and I'm really thrilled that they can present here for you today. So without further ado.
Good afternoon. Thanks so much for being here, either in person or virtually, and thanks for the wonderful introduction, Deanna. And Margaret, for all of your help with the logistics and setting things up. So I'm kind of used to having a typing thing right behind here. I guess I'll stand here and look at this. That way I can look at you at the same time. So this is Janet and Rob's version of the same title. Mine is the Succinct Conservation Challenges. And there's a couple of things that we're going to reiterate multiple times today. This animal been listed as endangered since 1997 on the federal side, and it is extremely, extremely endangered. So any adjectives you'd like to use to describe it would apply. And it's an animal that's right on the brink. So what I'm going to do is give you an overview of its natural history, some of the ecology that we know about it, some of the ecology gaps that we really don't know about it, talk about our work on disease ecology, genetics, and current status. Then we'll move into Rob's material, and then kind of summarize in the end what are our projections for the sustainability and the status of the species, and also what can be done potentially to make it more sustainable. This is a slide courtesy of our lovely Sci-Aid, Austin Ray, who's sitting in the third row here. And what he did here is he compiled a bunch of historical landmarks over the history of the Amargosa vole. If any of you have been down to the Shoshone, southern Death Valley, Tacopa area, it's an area very rich in history anyway. There's the not the gold rush, the early miners, the 49ers, and I'm sorry, I'm misspeaking. The borax teams, the mule teams and borax, and a lot of mining, and early Old West stuff in that area. And so there's an interesting museum in the town of Shoshone that tries to assemble some historical artifacts from the Paiute people that were there originally through the westward movement of people in sort of around the time of the construction of the railroads. But going even farther back, 10,000 years ago, this wasn't a desert. This was a wetland. It was a wetland throughout much of the entire area, and that would have been the late Pleistocene. And at the end of the late Pleistocene, the area started to dry. So people always say to me, how could a wetland obligate species be in an area like this? Well, it didn't used to look like this. And we'll show you in a second how small the fragments can be, the little refuges where this vole and other wetland obligate species now are constrained. But as the water receded and the area became desert and more isolated, the vole became so separate from other nearby subspecies. The vole was first discovered. You're going to have to go up and then down because the first one is 10,000 years ago. Then we get to 1900. The Amargosa Vole is discovered through UC Berkeley researchers coming through the area and just cataloging everything that was living in the area. 6 years later, the Tonopah Tidewater Railroad came in, and that had a pretty profound impact both on trade, also the connectivity of the people with areas outside, but also in changing local microenvironments because areas that might have been two contiguous marshes are now going to be split into two on either side of the railroad. It creates places where water no longer can flow or land may back up against the railroad. So there's a real concern that the railroad may have changed the area quite a lot. In 1917, according to Tracy Storer's records from the Museum of Vertebrate Zoology at Berkeley, the vole was extinct. He went through the marshes in both Shoshone and Toccopa and couldn't find any. 1936, rediscovered. And then kind of skipping ahead in the 80s, thought to be extinct, rediscovered. So all of this says to me that even for 100 years, this can't be an animal that's been widely ranging or highly abundant, because if it were, people wouldn't have thought it was going extinct. And it's actually only in the really modern era that we start to get a good handle on exactly where it is and exactly how many there are. 1967, I think, is a really important landmark. That is this one right here. And there is a artesian well that was dug in the desert, and it came up with quite a lot more pressure than was anticipated. The water did, so it actually persists today, and until 3 years ago was the best quality wetland, even though it was man-made, for vole habitat. And so that's from 1967, and it created a really lush oasis in the desert out of a spring with quite a few hectares of dense green bulrush. 1980, the state recognizes that the animal is endangered, and this as such. 1984, the Fish and Wildlife Service does so as well. 97, 98, the recovery plan from Fish and Wildlife Service is published. And it's a good document, but it's based on a lot of questions because not very much was known about the biology of the vole. I was brought in 2010 by Deanna and Tammy Branston, a biologist in Region 6, to look at a disease problem that was discovered on the vole's ear, and I'll cover that in a second. 2011, we went, well, we need to expand beyond disease and start thinking about the entire demography and biology of the species. And 3 years later, we're continuing on with demography, occupation, population viability analyses, disease, genetics, kind of whatever it takes to get the job done. Maps, love maps. This is a map, as you might guess, of California, Western Nevada, Southern Oregon, and Northern Baja California. And the California vole, sorry about this, the California vole is a common species. In fact, in a lot of places where the California vole is present, it's considered a pest. And there are a number of subspecies of California vole. In fact, there's 17. And up in this part of the state, all of those subspecies coalesce or have areas of overlap. But as we get out into this area that I was just talking about, that Pleistocene water area in the Mojave Desert, the voles are now in very, very isolated populations that have evolved to become distinct subspecies. And our subspecies of interest is, sorry, I'm having a little bit of issue getting the mouse to be on the place I want it. It's right here in far eastern California, not too far from Las Vegas, and the scientific name is Mycotis californicus stripensis. Here is another view, a Google Map image showing the Amargosa River drainage. The Amargosa River is kind of a unique river, and Rob will probably talk quite a lot more about it. It originates here, goes south, takes an abrupt north turn, and dies in Death Valley. And across extent of the Amargosa River. Occasionally it's above ground, and a lot of the time it's below ground. So where it's above ground in these little fits and starts, in these little pools or oases, can be habitat for the Amargosa vole. And finally, to put us on the map, this is the ecological setting, a map from Rob, showing the entire Mojave ecoregion, which if you go into the literature, actually people argue exactly what is the Mojave ecoregion, whether you're basing it on indicator plant species or what have you. But regardless, again, we are here, right around there, still within California, but basically right in the center of the Mojave. Here's a nice picture. I don't know what the lights are like. Are they okay for you guys that are here? Are you able to kind of see any of the detail? Okay. What I wanted to show here is the first picture of Amargosa vole habitat. And in this picture, what I want you to appreciate is, first of all, this is the borehole, that man-made pool, and you can see steam coming up off of it. It is a hot spring. Parts of it are very hot. And there's a very narrow fringe of vegetation, primarily bulrush, which is the bull's main place to live and food to eat. And outside of it is this dry, alkali hardpan. And, you know, it's 120 degrees. There's absolutely no plant growing in it. And any bull that would step on it would burn his toes, but actually, more importantly, have to go right back into that vegetation. So in between these pools, there's nothing that the bull can use at all. And, you know, sometimes people will challenge us and say, well, other than being isolated, what makes an Amargosa vole special? It's not all about me, but one of the things that makes them special is how much I love them, and you all should too. But in addition, there are some biological justifications for what makes an Amargosa vole an Amargosa vole. Original description said they had a narrow zygomatic arch and a lighter colored pelage. What we see today is a really large California vole, sometimes a third again or twice as big as California voles from Northern California. They will bite, of course they'll bite, but they bite a lot less than their other California voles, and they're so docile that it's led us to ask, is this an inherited trait, or is there something about their food or their environment that they're so docile? They do live, nest, burrow, and feed in bulrush. And all the ones we see are generally about the same color, a mouse brown color, but what's I think new is there's a phenotype, probably driven by the alleles of a little white mustache, and 90% of the animals have this mustache. And that's an interesting fun fact, but it also reflects the fact that they've lost virtually all the detectable genetic variability that we think that they used to have. And so even maybe insignificant alleles such as those that code for a white mustache are now fixed in the population. Status of the species. Boy, that is probably the million-dollar question right now. And then the next one is what to do about the status. The status could barely be much worse and still be in existence. We're going to argue whether there's 80 or 100 hectares total. What are you saying, Rob? No, we're not. We're going to say there's about 80. I mean, even if we argued about it, this is an incredibly small amount of habitat for an animal. And even those 80 hectares are not connected. So what you'll see here on this side are a bunch of disjunct marshes, and outside each marsh is that dry alkali hardpan. This, for example, this is borehole. It's a little uphill. So, but an animal that lives in the borehole patch, I love your mouse, just love your mouse. An animal that lives in your borehole patch has to, thank you, cross the road. I can't do it, whatever. I'll just spend all my time messing with technology. So if he's up in that red patch up there, he has to cross the road. He can go down to the patch across the road, which is also red. It's red because we have seen bulls there in the last couple of years. He can go out across kind of a wet marshland area with inappropriate vegetation and maybe hit a couple other patches, which are now gone. So you see kind of a pattern of uphill patches where there are some bulls, maybe 2, maybe 10. Borehole used to have more. And especially if there's flooding, the animals can end up down in the downstream marshes. Few of the marshes are not particularly accessible most of the year, so we don't have really good data yet on exactly whether there's bulls there or how many there might be. But regardless, we know that migration probably goes mostly downstream, and so we don't really have a true metapopulation of animals coming and going. Each subpopulation is not sustainable. There are a few that probably are, but they're so small now in numbers that they wouldn't really be considered a sustainable population. And while the recruitment or the ability of these rodents to recruit juvenile rodents is very, very good, they also have extremely high mortality. So the other day I'm making this slide, and I have a 21 and a 15-year-old kid going, oh my God, that is so bad. And so I took their advice and made the slide their way. So here we have it. What I'm showing you here, we will start in the marsh. And remind me, I'm okay camera-wise if I'm here? No. Here? I can't move it on here. Well, he's going to make it better for me. Thank you. All right. Green stuff in the middle. The most important plant to this animal is Skinoplectus, which is bulrush, and has been named. It used to be a scopus. So it's a bulrush species. And there are a few other plants in the area, but as far as we know, the bulls neither eat them nor nest in them. Something to be decided. So we've got a couple of plant species graphically represented there. And within the marsh, we really only have two rodents. The larger Amargosa Vole and the smaller western harvest mouse. So they largely do live completely sympatrically. Outside the marsh, right on the outside, in the plants, we start to see wood rats, house mice, which are non-native, cactus mice. Did I forget somebody? That's pretty much all of them. And there's an opportunity for spillover disease or competition with those, but not really directly because they're not in exactly the same place. But of incredible importance is this vast array of predators. How many predator species are we thinking there are now? 30? 30-ish? So I represented a raptor, but we've got prairie falcons, we've got northern harriers, we've got potentially golden eagles. We've seen them there, that they would necessarily take a bull. We've got a whole bunch of bitterns, egrets, a couple species of egrets, herons, and so forth. We have lots of coyotes. Every night in the year, you hear the coyotes sing. A couple species of snakes, bobcats, domestic cats, unfortunately, coming in from local areas where people have had cats, and then owls. So those are everybody's favorite snack. These are some cameras, pictures from Reconics, remote cameras that have been set in the habitat. And partly I'm showing you this because they're really cute. And also I'm showing you because I like the birds. All of those birds, oh yeah, I'm in the wrong place. I'll stand for it. All of those birds are going, yum, tasty. And that's what they do. The bitterns sit there right, thank you so much. The bitterns sit there right, almost camouflaged up against the blades of the bulrush, nice and tall, and then they just stab those bulls one after another. There's a harrier that sits right over a bull hole. Every time I'm there, the harrier, I figure, how many bulls does a harrier eat a day? One? And there aren't that many bulls in borehole. Okay. So as Deanna mentioned to you, my main field is disease, and I think disease is playing a role in making life harder for these animals. But as a disease ecologist, I think, to be honest, one of our first questions is, just because we know that there's pathogens present in the ecosystem, what is the significance of those pathogens to the population that we're interested in? So that's a question we've been asking ourselves. And there's a couple different ways that disease could be a problem for a endangered small mammal. Pathogens, when they come in and make animals actually sick or die, have a role which would be analogous to predation. It's removal of an individual, sometimes the weakest individual, from the population. So even if there's no chance involved, a disease can come in, harvest a few animals, but usually the population would recover because it's just another mechanism that keeps them below their carrying capacity, with a few exceptions. One is frequency-dependent transmission, which I don't really have time to talk about, but it's a real issue, but it has to do with the fact that even as the animal becomes really rare, the pathogen doesn't go extinct. Sometimes diseases can impact one species more than they impact the other species, and then that's called it's a form of apparent competition. We think the two species are actually competing, but they're not. What it is that one is more vulnerable to disease. And then, so tactically, we can have diseases that do things like make it such that by chance, if the animal gets disease, they're less likely to breed or more likely to die of predation. And then finally, we can occasionally have catastrophic epidemics. We were concerned that that was the case with the mite, that this might be a catastrophic epidemic. As it turns out, it seems that the mite doesn't have a huge fitness cost to the bull, but we have other evidence that there's other diseases on the horizon that may be. So, disease, there's not probably any disease that I can think of except the mite that's likely to specialize on the bull in this ecosystem. Most of them are going to be pathogens that are spilling over from another host species or maintained by multiple hosts in the community. So here's a list of organisms that were either screened for by a master student that Deanna and I co-advised over the last couple of years, Caitlin Otcom, and she went out and surveyed for these different pathogens in this community in Tacoma, or there are pathogens that we went, well, we need to start thinking about these guys in addition to the ones that Caitlin looked at. I'm going to talk about toxo in a second, but notice that 13% of the animals, the voles, had evidence of toxo infection. Borrelia burgdorferi I'll talk about. Anaplasma is a tick-borne pathogen. I doubt it has any relevance to bulls at all. Rickettsia as well. Relapsing fever would be a human pathogen. Ticks, fleas, mites. Then we'll talk about hantavirus, Yersinia, and Francisella. Those are all human zoonotic pathogens, and it would be important for us to know, both for the people who live there, but also the people who work there. Hantavirus, we're talking about Syndrome Bray, and that is a very dangerous pathogen that's transmitted by small mammals. And today, all of our data suggests that there's no hantavirus in this patch, even though this is in Yeo County, which has the highest prevalence of hanta in the state. Yersinia pestis is the agent of plague. Voles are absolutely known to carry and potentially be reservoirs for plague of California. Again, no evidence of plague in this patch. Francisella tularensis is the agent of tularemia. And that would be, of all the pathogens I can imagine, the one that, if it were to come in, would have high mortality in the bulls itself and could cause a catastrophic epidemic. And finally, leptospirosis is another zoonotic pathogen spread by rodent urine, and that work is in progress. The orange. The phone call we got was, there's orange fungus on their ears. And it isn't a fungus, it's a mite, but it kind of looks like a little rubbery fungus. I'll show you a neat picture in a second where you can see there's multiple orange organisms on their ear. So the concern was, I believe it was fall, and quite a lot of the bulls not only had orange fungus crud on their ears were gone. And it's really hard to appreciate, if you look next to the forceps here, that isn't just what you can see in the picture. That's the only thing left on his little ear pinna. The rest of it has literally melted away from the tissue destruction and necrosis that was caused by the mite feeding. We didn't think that was healthy for an animal. And a greater concern was the fact that some of these mites are on genitalia. And our thought was, if that's going to cause this tissue damage in the genitalia, it might inhibit reproduction. And here's a neat picture. On the right, we have an electron micrograph. These guys are so tiny that we had to use electron microscopy in order to see enough features to identify them. And on the left is a little cluster of chigger organisms all feeding together. So when we get up on a compound, light microscope, and see all the chiggers together, that's what they look like. We did some DNA sequencing, did some examination of the morphology of the mite, and its closest relative, and probably what it is Neotrumbiculum microti. And the trumbiculate mites are the same types of mites that cause scrub typhus. They're chigger mites. As larvae, they're obligate parasites. The microti specific epithet suggests that it's a specialist on voles. Not in this ecosystem, it had never been described before, but I'm fascinated by that because it suggests to me that it's related to a mite that's in voles in other places that are much less close to this habitat. Okay. And let's look for a second at what chigger mites do. Look at the cartoon on the top. that's a cartoon of a chigger mite attached to a host skin, and it's feeding. And so what it does is it puts its mouth parts into the first part of the skin, and then it sounds like something out of Star Wars or Star Trek or something. It starts spitting out digestive enzymes, and those digestive enzymes create a feeding pool in the skin, and then it uses its mouth parts to suck the digested liquefied skin material back out. And so put enzymes in, suck, enzymes in, suck. And the host responds, so this is a light micrograph histologic section of a host response. On the top, you can actually see mite mouth parts. So those little bunny ears on top are mite mouth parts. And then we come through the skin and we see that area just kind of open. It looks like the inside of a Slurpee straw. And basically, a Neotrombiculum microtes thinks of a vole as a giant Slurpee. So it does induce a lot of inflammation, necrosis, reactivity, itching, pain. We'd have to ask ourselves why voles are so severely affected. We have found this mite on a few of the harvest mice, not as many, but the harvest mice. The harvest mice don't have any of these. They have the mite, but their ears are fine. So, we'd have to prove this. But my number one hypothesis is that genetically, the voles have alleles or have lost the ability to really resist this pathogen, to resist the severe effects of this pathogen. So, cutting to the chase on our disease work, one of the things we're aiming to do, we're seeking money right now, is full genome sequencing to look at disease-resistant alleles in Amargosa voles and other outbred voles, to see if these animals still have the capacity to respond to diseases that are going to be reintroduced into their area. We haven't really documented too much of a fitness cost from the mite on the vole. We were concerned that it would reduce grooming, reduce feeding, reduce reproduction, and some chiggers do transmit disease, like I mentioned, scrub typhus. So all of that is still underway. Disease number 2 of significance is toxoplasmosis, and many of you probably know that toxoplasmosis has a definitive host, which is the cat or felid. Both bobcats and house cats can transmit toxoplasma sexual stages, and when they do so, it contaminates their feces. Small mammals are secondary hosts for toxoplasmosis. The agent insists in their muscle, and let's say human beings or other animals could acquire toxo from 2 different routes. They could acquire it by either eating infected muscle meat, or they'd acquire it through exposure to infected cat feces. Well, that's fine if we're worried about us, which we are, but if we're worried about the voles are going to acquire it through exposure to cat feces, and it does insist in their muscle. And when I went to vet school, we were taught it insists in a mouse or a small mammal's muscle, and it's not a problem. It just stays there. The only problem is the likelihood that it may transmit over to something else that we care more about, like us. This is not the paradigm anymore. What we recognize is that toxoplasma has the ability to manipulate small mammal host behavior in a way that's really clever. Small mammals, when infected with toxo in their brain, have a behavioral change that isn't just they start seizuring or they start to have abnormal behaviors. They become attracted to cats. And this has been shown with both rats and mice, and we're aiming to show it with voles. If you were to take an infected small mammal and a cat urine soaked thing, a sock, let's say, most small mammals, when smelling cats, would leave, and infected mice and rats, when smelling this object, are attracted to it. They go to it, they rub on it, they do all the things that would actually promote transmission of the infection. So it's a very complicated, evolved response on the part of the pathogen to ensure that the secondary host and the primary host contact each other. What's scary about that is the possibility that this pathogen could manipulate vole behavior in such a way that the voles can do something that will reduce their numbers even more than they are. Like I said, we're aiming to show that with experiments right now with another one of the students in the lab. The last disease I'm going to talk about is tick-transmitted disease, and this is an Amargosa Vole, and you can see his little white mustache. But you can also see that he's got a fairly prominent tick on his little soul patch. Isn't that what that is? This is cheating. This is not an Amargosa Vole. This is a coastal California vole, but it's kind of cool because he's totally loaded with ticks. Well, we took him off. So, what's the significance of ticks? Are we worried about Lyme disease? No, not really. In just 2011 to 2013, we found 62 infested Amargosa voles and a fair number of infested harvest mice and house mice as well. And every tick that we found was Ixodes minor. And I'm pretty sure that almost all of you are not really familiar with Ixodes minor. But Ixodes minor is in Central America and the American Southeast. It is not in the American West until now. I'm working on dating when it first came, but it may have only arrived 3 to 5 years ago. It almost certainly came on birds, and in the American Southeast, it is expanding. It's expanding in range and numbers. And it is a competent vector, for the agent of Lyme disease. And so in the American Southeast, northern Florida to Virginia, it's considered actually quite a concern because it doesn't bite people, but it does feed on small mammals and birds, and it probably will maintain and actually elevate the prevalence of Lyme disease in those areas, allowing the human-feeding tick to feed on these small mammals and then feed on people. That's not our main concern here, and I don't think that Ixodes minor itself will hurt Amargosa voles, nor do I think that the Borrelia that it's carrying will really hurt Amargosa voles. But I do think that it suggests to us that there's a route by which birds can, and probably other species, can continually reintroduce pathogens into this very isolated ecosystem. And if they do so, we've already demonstrated, we think, reduced genetic ability to respond to pathogens, and a population that's so close to blinking out that it really can't afford too many more challenges. So we did a lot of DNA sequencing work and electron microscopy to demonstrate that this really was a tick that was not native to California and had never been described in California before. Convinced ourselves, the very skeptical health department, the Georgia Southern tick specialist, so finally everybody says, yeah, it really is. Wow, that's amazing. And all I want to show you on this phylogenetic tree, let me do this for 1 second, is here is Ixodes pacificus, which is the tick we thought it was. That's the common tick in California. And Ixodes minor is, 5 lines down. It's actually not very closely related to the native tick at all. Ixodes minor was known to be from Central America. It has expanded in the American Southeast, as I mentioned. It does feed in all of its stages on small mammals or birds. Okay, and people say, well, how could it possibly get to the middle of nowhere, south of Death Valley? And the only way I can imagine, obviously people or dogs, dogs wouldn't even transmit it because it doesn't feed on dogs. Really, the only way it could have come is on migratory birds. We have been, if people ask, have you been doing birds? Yes, 2 springs in a row. So far, I haven't gotten them. This Ixodes minor, as a fun fact, I don't want to belabor this, does have a Borrelia in it. Borrelia is related to the agent of Lyme disease. This Borrelia is not truly the agent of Lyme disease. It's a close relative. But where is this Borrelia from? Guess where? It's from Georgia. It came with the tick from the expanding patch in the eastern America. And just a tree to show you its relatedness to Lyme disease. So our disease work does ask us to sort of, stand back and say, what is the importance of this to the conservation of this species? Well, I feel that ecosystem health as a bigger picture is always something that we want to understand our conservation work as part of. Also, we have to ask which of these diseases can actually regulate this population. Toxoplasmosis almost certainly could. Tularemia, if it were to come, could. And the others are still a little bit of an unknown, the mite, for example. If disease resistance, if inherited disease resistance is impaired, and we can show this, what will happen as newer and more novel pathogens are introduced? And when we do, if we do, captive breeding and translocating, how do we manage for disease? Because are we cool with just taking diseases to new patches? Or do we leave the diseases behind, which means that there won't be any ongoing process of coevolution between the host and the disease anymore, and ultimately the host loses? All right. I don't want to represent this work as my own. This actually is from Chris Conroy, who is the mammal curator for the Museum of Vertebrate Zoology at Berkeley. And he is a vole genetic specialist, which is a thing. And he's even into it more than I am, if that's possible. And it's a little hard, I think, to see the map on this presentation, but I just want to point out 3 really interesting facts. Fact number 1, the mitochondrial DNA and the genomic DNA come from 2 different lineages. That's weird. How'd that happen? Well, it happened because their ancestors broke off in different directions with the Pleistocene water loss after the recession of water at the end of the Pleistocene, leaving behind mitochondrial lineages from one group of voles and then the full genome paternal lineages from the other. And then the other thing I want to point out is that there's very, very little genetic variability in Amargosa voles. We've looked at multiple microsatellites. 5 of the 12 show no difference, show a single allele altogether. The others show 2 alleles. In Marsh number 1, our until recently largest population, there was no detectable genetic variability at all. We can't use genetics to infer metapopulation structure because there's no variability to do so. And so it's okay. There are other species out there. The northern elephant seal is a good example of animals that persist in the face of extreme loss of genetic variability, but it's not what we ordinarily want. So we need to define the status. That's why we're pushing towards full genomes, and better understand possible loss of variation that's needed for fitness. Okay. And I think that put you up, Rob? Yep, that's the switch. And I'll be back.
Okay. That was a great introduction. Really good introduction. And what I'm going to be doing is focusing on the links between population dynamics, demography, and habitat selection. Now, the conventional wisdom has been that the Amargosa vole is tied to bulrush, but if you look at other subspecies of Microtus californicus and other species of Microtus, they commonly occur in grasslands. So a huge question was, why aren't they using these extensive stands of salt grass, which actually makes up the majority of the vegetation in the wetlands out there? Or is it simply a matter of we haven't looked for them there? But before I get into all of that, I want to take another step back and look outwards a little bit and really try and give you a feel for what the setting is that the vole occurs. Janet already showed you it's isolated. We're going to look at that again. It's also extreme and it's variable, both seasonally and interannually. But just because you have isolation and extremity and variability, that does not make it a closed system. And Janet just did a really nice job showing that with ticks, you can see how biotically it's an open system. And what matters from a habitat perspective for the vole is it's also quite open in terms of recharge of water to the marsh. There is local recharge from springs, but the bulk of it is coming from distant mountain ranges. That's a really key point to what's happening now for the last 9 months. So again, just reiterating, if you take an eco-regional view, you could not get more isolated. It occurs right dead center geographically in the Mojave ecoregion. But there's an environmental significance to that too. This other project that I'm involved in, looking at climate and fire regimes and vegetation dynamics and non-native invasive species throughout the Mojave ecoregion, One of my collaborators on that, Jerry Tagstad at the Pacific National Lab up in Washington, has done this really nice analysis. And he's shown that the Mojave has 4 major precipitation zones. And they're defined by the amount of winter or monsoonal season precipitation that comes in. And all of that blue that you see here is what we call the low winter and low monsoonal precipitation. And I will give you one guess where that above ground portion of the Amargosa in California is and where our favorite vole is, right in the middle of the blue. So it's not just the isolation geographically, it's this extreme setting. It's the driest part of what is already one of the driest ecoregions in North America. Temporally, keeping with the water theme, I've been analyzing data, time series 60-plus years, on the pattern of precipitation in the Mojave. The red line simply shows that there has been no trend, but this is the key one. The black line, it shows how variable it is. It's variable between years, it's variable between decades, and that's going to influence the habitat of the vole. And you get a sense of the seasonality by simply looking 6 months apart, more or less, at the vegetation. It's not green all year round. The green up starts late March, and it peaks right about now, and then by August, it's pretty much starting to go into dormancy, and it browns out. And, of course, this has an effect on the vole's habitat use, presumably. Okay, now let's start looking back into the vole. This idea of distribution, abundance, habitat selection, these were fundamental pieces of information that we had no data on whatsoever, but maybe a little bit on distribution. And the habitat selection, well, it was a little bit biased, and certainly no really good numbers on abundance. But those depend on how you look at them relative to the scale. So that's how we were trying to think of structuring our study. We only had enough funding for 6 months, so we really wanted to try and get it as right as we could with this limited amount of time and funding that we had. So we figured to help us think through this, let's come up with a conceptual framework. And what really worked for us is thinking of the vole's potential distribution and abundance along an environmental axis and a geographic axis. And you could start coming up with different potential scenarios. Now, if it was like this wouldn't be so bad. We would probably be less concerned about it. It's got broad niche breadth. It's got a pretty broad geographic space, but there's always trade-offs in this kind of arrangement. And the trade-off here is if a disease did get into the population, transmission would probably be enhanced considerably. So we can just run through a few different scenarios, some variations on this. Narrow niche breadth, broad geographic distribution, and a contiguous population. And then one more, same idea, but it's disjunct, it's patchy. And then you can flip that around, and you can say, well, they have broad niche breadth, but they're limited geographically, contiguous populations, and the disjunct, patchy version of that. The nightmare scenario would be this. Limited in niche breadth, limited geographically, all living in one place. If it was like this thing is screwed. It's over. It does not have much hope at all. And that's what we were hoping we would not see. Now, given that framework, we could start asking some questions. What's its habitat? What condition is that habitat in? How's the population structured spatially? What are its temporal dynamics? And what kinds of stressors are on the population? We already knew that this infection disease was a potential one. What other ones are out there? So we can start pulling these questions into that conceptual framework. If we're going to ask what are the relative contributions of these demographic variables, that's a question of temporal dynamics. If we want to know what the distribution of abundance in the population is, that's more a question of geographic space. The relationship between disease, infection, and demography, that's one of stability. Looking at that from the habitat component of the study, if we want to know how closely voles are tied to bull rush, if we want to know what exactly is vole habitat, those are questions of environmental space. But if we want to map the environmental space onto the geographic space, that's a little bit more tricky because now you're dealing with both axes at the same time. So this is where our thinking was as we went into the design of the project, and we took a fair amount of time before we went out in the field coming up with, a decent design relative to the resources that we have. And we had 6 randomly selected grids. These are 1 hectare in area, and these were going to be our major large sampling units. These were randomly located, and they were also randomly oriented, and that was so we could capture the patchiness of the vegetation out there and avoid the self-fulfilling prophecy that voles occur in bulrush, so we're going to trap in bulrush, and then we'll find that they use bulrush. We want to do a little bit better than that. So we trapped along about a 3/4, maybe a little bit more than that, section of the watershed, and the spacing between the grids ranged from about 75 meters to around 250 meters. Now, if we drill down into one of these grids, we divided them up. There were 108 traps, and we divided them up into 12 subgrids. These were 225 square meters in area, and we did this specifically so we could look and see how habitat was affecting local occurrence of the voles, relate the two together. We also did it so we could measure movements within this local scale. So each of these 6 grids was trapped more or less simultaneously for 5 consecutive days for 5 months running between January and May 2012. For the habitat work, we took some fairly detailed measurements. We took these at both the trap and the grid scale, which allowed us to come up with estimates of vegetation species composition, gradients, turnover, habitat structure at the trap scale, which is about 1 or 2 square meters, the subgrid scale, which is 225 square meters, and the grid scale, which is about 1 hectare. And we did the data collection both at the end of the winter and then towards the peak of the growing season, the latter part of the spring. We also wanted to get a sense of how much habitat there was out there. So Michael Cleaver, one of our biologists, he pulled down some really high-resolution NAPE imagery. It stands for National Agricultural Inventory Program. It's 1 square resolution imagery. He did some very robust classification methods, and then I went out in January and February of 2013 to 600 randomly selected points and did what's known as blind ground truthing to validate, assess the accuracy, essentially, of this map that Michael developed. Now, this is a very powerful design. It allowed us to look at the habitat question with less of the bias that had been there before, quantify the temporal and spatial dynamics in the population, and come up with these monthly population estimates. And probably most important, it enabled us to link the temporal and spatial dynamics. All right, so let's start looking at some of these data. We're going to start off looking at abundance. This is density, so individuals per hectare on the y-axis, and here's our 5 months of trapping. And what you see is data for 4 of the grids. Why isn't there data for the other 2 grids? Because we didn't get any voles there. We'll get back to that. But what really jumps out at you here is the huge spatial variability. The borehole, Marsh 1 that Janet was talking about, that 2-hectare marsh that's right at the very northern end of the voles range, their densities range between 10 and 40 individuals per hectare over the period of that study, whereas in the other 3 grids it ranged between 1 and about 3 or 4 per hectare. So a major change, major disconnect between different areas and how many voles were there. The other thing you see in this is, that population looks like it's increasing and then decreasing and then going back up. And that's exactly right. That is what it did. And we can express that, we can look at that as the population rate of change, lambda. So all lambda is the population now versus the population at the previous time step that you assessed it at. And between January and February of 2013, the population increased basically by 150% in 4 weeks. Not bad. It then remained stable, and then when the rate drops below 1, that indicates a decrease. So it seemed, and then they increased again between April and May. So it seemed like there was this potential for rapid increases, and the decreases, at least in this time period, did not seem quite as extreme. When we looked at abundance, relative to the cover of bulrush. So here's mean bulrush cover at the grid scale, so we're talking 1 hectare, and here's density. You see a rather dramatic increase in density where bulrush cover is greatest. But what also is very interesting was if you looked at the variation in cover in bulrush, how patchy it was. The density was greater where there was less patchiness of bulrush. So it's not just mean cover, it's also how that cover is distributed. Now you have to be a little careful because there's a statistical artifact in here. As you get more cover of bulrush, you're going to get less variability. And that was a relatively minor effect because of the way we sampled. It's there, it's present, but we were picking up a real biological pattern that it was constant across seasons. Okay, shifting into some real basic demographics. Here are the females, here are the males, and this is the proportion in adult, subadult, and juvenile age classes. And you can see that there's an even sex ratio and what I would call a mature population. The vast majority of individuals were in the adult age class. Okay, all that's fine and good. How are they recruiting? The per capita recruitment. If there's 2 of you now, was there 1 of you or 1/2 of you or 3 of you the previous time step? And what we see is a variable recruitment pattern. Okay, fine and good. That's interesting. It's actually fairly high, around 2 individuals in that period when the population increased between January and February. But what was also interesting, really caught our eye, was it showed the same pattern as lambda. So that was our first aha moment. We were wondering if that could be due to dispersal. If you look at this frequency distribution between the mean maximum distance moved by a vole that was captured on more than one occasion, what we found was they really don't move very far at all. The mean distance was around 25 meters. About 60% of the voles, they occurred in just one of those 225 square meter subgrids. About 1/3 of them inhabited 2 of them, and there were occasionally these long distance movements. But what was important was we did not document any instances of intergrid dispersal. All the movements were occurring within the grids, even the ones that were pretty close to each other. So we said, all right, this recruitment rate that we're seeing, it doesn't seem to be influenced much by any kind of dispersal, major strong dispersal effects. So we wanted to look at breeding and survival. What this graph is showing is by month from January through May, the proportion of females that were reproductively active. And this is what was lining up with the recruitment rate. When the population was low density or decreasing, then the proportion of non-reproductively active females increased. When the population was increasing or higher densities, you had a very high proportion or higher proportions of females that were actively breeding. Then there was the survival, and it was variable, and it's matched up with the recruitment rate very nicely. Then there was the survival rate. It was constant, but it was low. During the period of this study, if you were an Amargosa vole, you had about a 1 out of 3 chance of making it out of a month to life. Expressing that another way, if you went to, say, February with 100 Amargosa voles, you'd come out the other end with about 34 of them. So very high mortality rates. And was that due to disease? Well, fortunately, or the infection, I should say, at that time, it did not appear to be much of a factor. What this is showing is the proportion of, the proportion of individuals that we captured between January and May that showed infection. The first couple months, there were 15% that showed some sign. They either had mites or they had previously had mites. It was down to about 5 or 6%. You didn't need to do any kind of fancy analysis on this. You couldn't because the numbers were too low. We did at least look and see if the number of months that a vole remained in the population between 1 and 5 months had any relation to the proportion that were infected. It was basically the same. So that actually gave us a little moment of breath. But now we had our first key pieces of information on the population dynamics. And what it appeared to be doing was survival was influencing the level of abundance. Whereas you've seen both of these panels before, here's recruitment, here's lambda, recruitment was influencing the actual rate of change, and dispersal was not having much of an effect on this. And the population dynamics were occurring at a local scale because dispersal was not, the populations just weren't linked. So when you get into something like this, you start looking for evidence of density dependence. And we did not find any evidence of density dependence on the measures of body condition of the voles, but what we did find, both in the current time period and the previous time period, is that there was some evidence of density dependence occurring for the probability of a female being reproductively active. So when you get that, you say, aha, even though they are at low abundance overall, they're not a particularly abundant subspecies, there is evidence that they are bumping up against a carrying capacity. So can we estimate it? And we said yes. Even though we didn't have very many data points, we were able to do this in the classical best method of estimating what carrying capacity is, which is no population growth, no increase, no decrease. You simply relate the numbers now, or the growth rate of the population against numbers in the previous time step. And our estimate of that relationship was that the carrying capacity expressed on a density basis was about 30 to 35 voles per hectare. And this is really useful information, especially for modeling. Now, starting to make that switch back towards habitat selection, from the demographics and the population dynamics to the habitat selection. At the grid scale, the estimate of occupancy, in other words, if you went out and there were 50 hectares of vole habitat, how many of those hectares would have at least one vole in them? The estimate was about 0.50. In other words, 25 of those hectares would have at least one vole running around. And the colonization and extinction rates, the dynamics, were basically the same. So at that scale, it seemed like, well, it's kind of stable. But we wanted to see what the relationship of the occupancy, the proportion of area occupied, was to bulrush. And what this is the ratio between bulrush and saltgrass. So this vertical line here at 0 means that there's equal amounts of cover of bulrush and saltgrass. And grids with less than 50% cover of bulrush were not occupied, and it went up rapidly in terms of occupancy. Real quick jump, even though it's low numbers, as bulrush cover increased. Okay, so what's happening at the subgrid scale, that 225 square meter scale? About 1/5 of the area was estimated to be occupied by voles, and it fluctuated a little bit, not to a great degree over the course of the study. In other words, a large proportion of potential habitat is not occupied by the voles. And again, if we look at that ratio between bulrush and saltgrass and how that influences the estimates of occupancy, past that 50-50 mark, you start seeing quite an increase in the occupancy rate, the proportion of area occupied. And you can get a sense on how heterogeneous this environment is for them, where they're having to select. This is an interpolated surface that one of our biologists, Paul Mayer, did for us, looking at that bulrush-saltgrass relationship. And what you see is very homogeneous, non-patchy areas are where you get a lot of voles, and as it gets progressively more patchy, you get progressively fewer voles. So now we can start honing in on the habitat selection question itself. All right, if we look at the number of voles that are captured in a subgrid, which are these top two panels, or the number of traps, which are these bottom two panels, what are they related most to? One guess, for the most part, bulrush. This panel on the top left, right here, is mean bulrush cover, and this is the number of animals estimated. It's variable, but it was a predictor, but the stronger predictor at that subgrid scale was the patchiness. If you go down to a trap, an individual trap station, about a 1 or 2 square meter area, it's all about bulrush cover. 80% cover, you guys can probably see the cursor moving. I can't. But it's where that curve really jumps up In that little patch there, you had to have about 80% cover of bulrush, otherwise you weren't going to catch any voles in it. The other factor, though, that did enter into it was a weak predictor, but it mattered, was litter cover, too. So, all right, now we're starting to get a real feel for just how strongly this vole is tied to bulrush. We can flip that around. We can say, okay, that was looking at it from the vole's perspective. Let's look at it from the vegetation perspective. And this is one of those complex multivariate graphs. It's actually one of the best methods out there. The people kind of go, whoo, what does that mean? And it's got a ton of information in it. What this is, each of these circles represents one of those 72 subgrids, 12 subgrids, 6 grids. They're color-coded by grid. And what these arrows are the structural elements of the habitat, so litter depth, vegetation cover, amount of bare ground. These little triangles here, kind of scattered in, are the vegetation species. So what you're able to do is you're able to look at turnover in the vegetation species and see how that is related to these structural elements of the environment. So, in terms of habitat structure, this first axis here was vegetation height and litter depth. The second axis was vegetation cover. The first axis for the vegetation species themselves was, not surprisingly, this incredibly strong gradient from bulrush-dominated areas to saltgrass-dominated areas. Where are the voles? That's what the size of the circle means. Proportionally, larger circles had proportionally more captures of voles. And most voles occurred in subgrids that were dominated by bulrush, but also nestled down in here, kind of hidden by some of those larger symbols. A lot of those subgrids that had apparently very suitable habitat were not occupied or had very few voles in them. And then there were interesting cases like this. This guy's laying out here in this patch of saltgrass and rush. And this is one of those outline observations that's really nice for giving creeps to the general pattern. We got about 3 or 4 individuals in this subgrid the first couple days of trapping in February. They didn't show up the last 3 days of trapping, and they weren't captured. Nobody was captured in that subgrid any time after that. They seem to be able to move into these, or be willing to move into these patches, but they don't persist. So the habitat structure that the voles like are these tall, dense, homogeneous stands of bulrush with deep, dense litter layers, and we can look at this much less technically by saying, there are the voles, that's where they're not, that's what it looks like usually where they are, and that's what it looks like usually where they're not. Michael produced a really terrific habitat map, very high accuracy, 91% overall, 95% accuracy for mapping bulrush habitat. And the things that jumped out of this habitat mapping exercise was we came up with 107 hectares of wetland habitat, a little over 1 square kilometer. In the original recovery plan, they estimated there was 20 square kilometers of critical habitat, so a little bit of a disjunct there. But what was more important is, of that 107 hectares of wetland habitat, only 30 hectares that was bulrush habitat. And a lot of those patches, and Janet mentioned this already, were pretty small. So if we bring this back to our conceptual model, this is what we were looking at. So what could we do with this information? Well, we could finally come up with an estimate of the overall population size. And we did. Over those 5 months, we estimated, give or take 15 individuals or so, that there were between 75 and 225 Amargosa voles in the world. Kind of a precarious situation. At least we had a number to tell us how precarious it was. We could develop population models that had both this explanatory and this forecasting function, and we could outline some preliminary management actions. These could be hypothesis-driven, so we could keep it within that adaptive management framework, and we could start addressing both short-term and long-term management scenarios. So just as an initial population model, mainly for explanatory purposes, what indicates is driving the population dynamics? We're going to extend our data here a little bit, do some simulations. So we identified 4 main subpopulations. We had an estimate of carrying capacity. Both sexes were in the model, adult, subadult, juvenile stages. We ran it for 20 years at monthly time steps. We were able to incorporate demographic stochasticity into it, environmental stochasticity, that evidence of density dependence, as well as this other type of density dependence called an LE effect, which operates on very small populations. We included dispersal, very low rates. Janet talked about this downstream effect, and that's what dispersal was related to, a flood. Maybe voles can surf. They can surf from one patch to another. So this chance of a 25% chance of a flood over every 5 years. And we did 1,000 simulations. The first thing that we found was a little bit of comfort. The model-based form of density-dependent lined up nicely, at least generally, with what we had observed empirically. So that was evidence that the model specifications were at least reasonable. But it also showed that some subpopulations are so small, where'd you go, little arrow, that they're never going to quite reach a positive growth rate. These are your sink populations. They depend on rescue effects, which are real problematic when you got a critter like a vole that doesn't move very far. And then as far as the sensitivity of the model to varying different demographic variables, we expected 1 or 2, maybe 2, to be important, but 3 actually turned out to have similar importance. The survival of the juvenile and subadult stages and the fecundity of females, all intuitively making sense. All right, there were still some lingering important questions. Why the extreme habitat specialization? When there's so much saltgrass out there and it's pretty thick, why aren't they using it? What were the main mortality factors, and were these related? Okay, so we lined up some potential explanations for this. In terms of microclimate, I went out in the peak of the summer of 2012 with a bunch of thermometers and measured the temperature in these thicker patches of saltgrass and these thick patches of bulrush, and they really didn't vary all that much. So we started to rule out microclimate. Food resources, they're herbivores, a lot of greenery out there, so that didn't seem real plausible, but that's food quantity, if you will, and we didn't know about food quality. So we couldn't entirely rule the food resource angle out, although we didn't think it really made a lot of sense. There was no evidence for competition. And this idea of lack of access to alternative habitats had no credence because the vegetation is all juxtaposed with each other. They had lots of access to alternative habitats. But Janet set the stage for this. How about those predators? And this is where we started to develop our hypothesis, this idea of a top-down effect. You have this 75 to 100-gram furry burrito running around out there that everybody wants a chunk of. And hey, I know Janet thinks they're cute. I think that they are a little bit limited because they can't avoid all these predators. We came up with a list when we were doing our work of about 24 potential predators. Sounds like it's expanded by about 25%. And we actually witnessed 3 predation events: American bittern, great blue heron, and coyote. So our hypothesis was that their habitat selection is being influenced primarily by predation, this top-down effect. But their population dynamics are probably being influenced more by food, quality, and maybe even over time, predictability of food resources. We still need to test this. This is just a variation of what's known as the chutes and ladders concept. So this gave us a roadmap into another year of activity. Our funding had run out, but Janet and Deanna had gotten some Section 6 funding, and so they're going to start studying the predation effects more closely. They were going to start not just trapping, but looking for sign in other marsh to see just what evidence do we have of voles being in these bulrush patches throughout their range at some time. And they were going to logically continue trapping at the borehole, the population core. I was going to continue to do a little bit of habitat sampling at these irregular intervals just to kind of look more at this relationship between bulrush and saltgrass, improve our understanding of the habitat relations as well as to inform some future habitat manipulation activities. Well, things change rapidly. I went out in June of 2013 expecting to see bat when I did the vegetation sampling at the borehole. And that's what I saw. This low stature but green marsh was now this low-statured, brown, and clearly dying marsh. There's virtually no emergence of bulrush leaves through that thick litter layer. And the question was, how is this going to translate to abundance of voles? So the trapping was done in November of 2013, and the number of animals that was estimated in that 2-hectare marsh was less than half of our lowest estimate from 2012, 8 individuals. And they were all confined in this narrow strip right around the flow out from that borehole. It's the only place there was green bulrush. Now, we couldn't come up with a true population estimate because trapping had only been done at the borehole, but we could make some very reasonable assumptions that were backed up by data, come up with a pseudo-estimate. And we guessed in November of 2013 there were probably about 3 dozen Amargosa voles left in the world. And all the evidence was that what was their core population had experienced a major habitat collapse. Why? Well, there's 2 critical factors for bulrush growth. One is warm soil temperatures and the other is standing water. There was no evidence at all that there had been change in soil temperatures. Fortunately, we collected that data. But here's a map of standing water depth. This is in January of 2012, green being deeper, probably on the order of 2, maybe even 3 inches depth of water. And this is about the pattern that you would expect in the winter. And then it expands as the water table comes up from recharge. This is June 2012. And then it recedes as the winter approaches. Then we expect it to go back to this. That's not what happened. That's where it went. That's June of 2012. That's June of 2013. That's November of 2013. Somewhere in that period, the recharge hadn't happened. And it's likely what has happened as the drying occurred. There just was not enough energy for the bulrush to punch through this really thick litter layer. The roots are starting to dry out. Bad scene. So now we were looking at this kind of situation. Our immediate concerns were that the situation had gone from precarious to dire. The poor core population was now likely a sink population. And any efforts at habitat manipulation, restoration, whatever you want to call it, had this very narrow window for them to be successful. And you have to be careful with statements like this, but I think most people are starting to agree that the Amargosa vole may now very well be the most endangered mammal in the United States. There were longer-term concerns, too. Marsh and the vole are really strongly linked. And the situation at the borehole had human influences as well as natural causes. There was evidence that other patches of marsh were also drying out, and that suggested a relationship with longer-scale climatic patterns. That was the situation going into this year, and Janet's going to bring it home.
But I think Rob gave you a sense that it's not funny right now.
We have probably one of the best collaborative groups of agency people, ologists, local stakeholders that you would ever want to work on a problem of this magnitude. A lot of the land is owned by BLM. Some of the land is owned by California Department of Fish and Wildlife, private landowners. There's the local Amargosa Conservancy, local Shoshone Village. People really, really motivated to protect this incredibly unique habitat, but also this incredibly rare animal. So that's actually some of the extremely good news. But first of all, we had to go from no data 3 years ago to this now. And second of all, everything we do needs to be data-driven, because we've never tried to do something this big and this hard. I think any of us, even individually or with our other experience. So we really wanted to produce as much data as quickly as possible and to give population viability analyses and time to extinction estimates so that managers, Fish and Wildlife, U.S. Fish and Wildlife, and Cal Fish and Wildlife can make the right choices. So right now what I'm going to show you are two different forms of population viability analysis. Sorry, I put my glasses up, I'll fidget. And we have a traditional one, which is the Lefkowitz matrix model that Rob described, and then we have a time series version, which has also been published, and that incorporates stochasticity and variability of growth. So the first one is the stochastic time series analysis, and I've given you the equation up here and the things that we include: time to extinction, starting with some initial population size. And it's a very simple equation with a lot of complicated math to back it up. The citation is solely 1997 in extinction models. But basically what we're saying, to the left I have a graph of California vole population time series. And these are over the period of time. And you can see these oscillations in their population size. And even at 0.5, there's really quite a high number, but the rest of the time it kind of fluctuates up and down. And our question is, if you have a very highly variable population, then the chances that it's going to hit these lows, these troughs, are particularly high. And if there's a lot of chances that it's going to hit those troughs, the chances that it's going to go extinct are also high. So alternatively, you could have, let's say you have a population that always is very, very low, but it's always very, very low. If nothing happens, the chance that that population is going to extinct is pretty low. So it's the variability in the time series that we're concerned about, in particular the times that it hits the trough. And that's even assuming things like borehole doesn't collapse, okay? So using a standard time series analysis and an initial population size, which we know for the Amargosa vole, a carrying capacity, which Rob just gave you for the Amargosa vole, and variance in population growth, which we don't know for the Amargosa vole. We don't have enough data. So where do we get any kind of an estimate of variance in vole population growth? We had to borrow it from other voles. We did compare it between multiple California vole studies and harvest mice. Because why harvest mice? Well, they're the only other animal in that habitat, right in the habitat. And actually, no matter where we look, California voles made better proxies for Amargosa voles than other species. But putting these numbers into this analysis, and it changes a little bit based on whether you change the actual estimated carrying capacity or the initial population size and not, but we're predicting a time to extinction of this species from 16 to 35 years, and a 10% chance of extinction in 2014. Full extinction, not borehole, the whole species. And that's the rosy scenario without incorporating the full collapse of marshland. Rob produced the Lefkowitz matrix analysis, which he introduced a few minutes ago. His main goal was also in this case to estimate the time to extinction. He's assuming 20 years. And that puts us out into the horizon of where the time series model was going into. And he can allow the conditions to remain as they have been, or he can allow the conditions to vary. But when he allows the conditions at borehole to vary and borehole to become a sink, obviously we have a much shorter time to extinction. And we're assuming that the other population carrying capacities across the patches stay the same, although there's evidence, possibly drought-associated, what have you, that they're not staying the same. They're also declining. So here's a couple of graphs. Please do step in if I just represent these. We just didn't want to keep having us switching back and forth our this thing. So here he's got no habitat loss on the top at borehole, and at the bottom we have habitat loss. There's envelope, 95% confidence envelopes around his estimate for standard deviations anyway. But the conditions, if the conditions remain the same as they were in 2012, then the population looks like it's going to persist. We've got months on the x-axis, and we look like either whether we have habitat loss or not, that we have persistence out on the top, but if we have habitat loss, a borehole down at the bottom, even if there is persistence, he's circling 1 and 0. And it takes 2 voles to persist, 1 male and 1 female. So that's not a number I'm very comfortable with. And looking specifically at the borehole population, same kind of thing. At the top, we have no habitat loss. At the bottom, we have habitat loss. And we've had the habitat loss. Despite my impatience to get water back in the marsh, how long do you take to get the water back in the marsh? They're tired of hearing this. I've finally been persuaded that the immediate crisis that drained some of the water was 3 years ago, and it's kind of already too late. We can eventually get water back in that marsh, but we have to reconstruct the vegetation in that marsh. Those animals are going to be gone by then. So the bottom panel shows what I just said. Predictions for the entire population, putting all the pieces together, 41% chance that the voles are likely to go extinct within 20 years, and if habitat loss is not reversed, they will go extinct during the course of the time that we're working on them. Predictions for lingering populations, again, if the habitat loss is not reversed, with 100% probability they will be extinct within 20 years. We didn't want to leave it at that, because you know what? We're not quitters, and the vole is not a quitter. It sounds so silly, but there are things we can do. We are obligated, Cal Fish and Wildlife and U.S. Fish and Wildlife are obligated to manage this vole. The landowners are as well and want to. The 1997 recovery plan, which was the earliest possible document to put together the problems, the ideas, kind of missed the mark in some places, but again, they had no data to work with, suggested that we should learn and that we should manage. They didn't put it this way, but this is how I've reorganized it. Let's document the genetic structure of the population. It's still in progress, but there's not much to document. Let's understand the basic biology of the species. I feel like we're getting there. Let's describe the extent of the occupied habitat, determine the habitat requirements, and determine the habitat quality. Bob just gave you a whole bunch of great data on that. We have graduate students, scientific aides, a great team from a lot of different agencies and universities working on all these, exactly these problems. Determine the population size. We did that. We probably 6 months ago would have agreed that we had really only an inkling, and I think in, what month are we in? June 2014, we have a very good sense. Outside the scope of the original plan, but we put it in there anyway, is all the disease work and population health. And then on the management side, Fish and Wildlife said, let's acquire land. They acquired a bit. There isn't actually that much more to acquire. I think there's 2 or 3 privately held patches that probably have couple handfuls of voles. There's not much left to acquire. Improve or recreate habitat, yes, that we can do. And not only can we do it in Tacoma, we can actually do it in the town north in Shoshone where they were first discovered and eradicated. We can reconstruct marshes and get them out of this one little handbasket and put them in other bigger places. Can we translocate, either supplement populations that are hanging on or actually create new subpopulations? Yes, we're starting to learn the technology to do that. And captive breeding makes me super nervous because we got, what if they all go extinct in a captive bred facility? I think we want multiple facilities. We want to know what to do with them when we get done with them. How do we get them to go back out and survive? So that's the focus now that we're sort of going, well, in the field it's really bad. What tools can we bring to make the field a better place for them? Recognizing that climate change is a real thing and is happening down there. Recognizing that the drought is a real thing and it's impacting them, and that they're going to have challenges. So they're going to need assistance, but I think we're trying to learn some of the tools to provide that assistance. And this is, what we would like to see. This is, unfortunately, where we're headed.
until we change trajectories, which we are planning to do. We don't think that the Amargosa Vole is particularly resistant. It needs what it needs. It needs bulrush. It needs water. But we are optimistic sometimes, partly because really on the politics side, there isn't a ton of politics. There's a bunch of people that have strong opinions, but they're really working well together to promote the survival of the species and the improvement of the ecosystem. Particular people are noted on the slide, but they'll be acknowledged in a second. We've been a little frustrated. Me, number one. I'm the least patient person on the planet, but our ecological data-driven needs are moving very quickly, but agencies take a while to respond, partly by law. And we've got a lot of hope because we know that the Amargosa Vole went extinct before, twice at least. And so if we continue to provide the level of support that is just kicking in over the last 3 years, I am somewhat optimistic. Just a beautiful, beautiful picture of the mouth of the Amargosa Canyon, a beautiful, beautiful picture of Borehole in its glory, and maybe not so beautiful, but in a way a very beautiful list of fantastic biologists and managers who participated in the team. And I know it's boring to watch somebody read their acknowledgments, but I do need to point out a few things. We've got heavy involvement from Cowfish and Wildlife, heavy involvement from graduate students and talented biologists at Davis, Berkeley. We've got plant scientists, animal scientists, we've got Fish and Wildlife Service, BLM, local stakeholders, and people just passionate about birds and wetlands and animals. And so far, a lot of the people who supported the work have put their money where their mouth is. With that, I will finish, and Rob and I will be happy to take questions. Thank you. Oh, wait, there's one more. Rob has a, oh, yes. Oh, it's not going to work, Rob. Yes, but they're not coming. They're not showing. Any idea? Go to next. Go to next. There we go. There we go. The weird USGS Vole boys. I mean, yes, that guy is in his bathrobe. And yeah, I think the USGS and Cowfish and Wildlife SciAids will say that they're working above and beyond their wage grade. And Rob wanted to thank our little friend, the Vole, for hanging in there. We happy now? Okay, thanks. And we will definitely be happy to take questions.
Okay, so any questions? So I'm going to pass around the microphone if you have a question, and that's so that the people on WebEx can hear your question as well.
What are the chances of captive breeding, and who's going to pay for it and do it?
Captive breeding has, I think, 3 big obstacles. One is paying for it. Number 2 is doing it. Just by putting voles in a room and saying, here's some food, now eat, survive, and mate is not that easy. And then the third big obstacle would be regulatory, right? We're not permitted to do captive breeding. However, Deanna and I did get funding for the first step of captive breeding in the next Section 6. And to start, Austin collected, how many, 10 Owens Valley voles as a surrogate. And they're at UC Davis right now. We're learning how to take care of them. They're picky. So far, their newest favorite food is carrots. The other grad student is going to try them on beets. And we're creating microcosms, mesocosms for them outside that are being planted with 3-square bulrush. So there's some work to be done between here and there, but we're going to, and I would have said 6 months ago, I was not prepared to do the captive breeding game, because we're talking you can get dozens of animals, not the hundreds or even thousands that we're going to need, but we need to. Who's going to pay for it? Initially, Fish and Wildlife Service and Cowfish and Wildlife, but down the road it's going to need to be a more sustainable money source.